The overall goal of this procedure is to generate induced pluripotent stem cells from human peripheral blood mononuclear cells obtained from frozen Buffy coats using a cost-effective and virus-free protocol. This is accomplished first by the isolation of PBMNCs from Buffy coats after whole blood centrifugation without a density gradient, followed by expansion in a specific blood culture medium. The second step is to transfect the Buffy coat PBMNCs with episomal plasmids by electroporation. The transfected PBMNCs are then plated onto fresh mouse embryonic fibroblast feeder cells. The final step is to manually pick induced pluripotent stem cells from the culture and transfer them onto new feeder cell plates for expansion. Ultimately, the efficient reprogramming of PBMNCs into induced pluripotent stem cells with a typical human morphology can be shown by immunofluorescence and measuring gene expression.
The main advantage of this technique over existing methods is that the generation of virus-free iPSCs from frozen Buffy coats reduces cost, working time and manual steps for operators during sample processing.
Another advantage to this technique is that it allows for the use of frozen Buffy coats stored in large-scale biobanks gathered from population study as a source material for iPS production.
Begin with collecting 8 milliliters of venous peripheral blood into a sodium citrate buffered plastic tube. Store the blood at 25 degrees Celsius and process it within 12 hours.